All right, welcome everyone. Today we are diving deep into the incredible world of micromanipulation. We're going to explore the super precise and honestly revolutionary techniques that have totally changed the game for assisted reproductive technology. If you're studying embryology or reproductive medicine, this stuff is absolutely essential.
So, here's our game plan. We're going to start by looking at where these techniques even came from. Then we'll zoom in on ICSI, what it is, why it's used, and its genetic side. After that, we'll go inside the lab for a step-by-step look at the procedure, check out some advanced methods for getting better results, and finally, we'll touch on other key procedures like assisted hatching and embryo biopsy. All right, let's get into it.
To really get a handle on today's techniques, we've got to go back in time. This isn't just a history lesson, though. It's the story of a clinical revolution that completely redefined what was possible for treating infertility.
So what you're looking at here are the real pioneers, the precursors to modern ICSI. You had subzonal injection, or SEI, where sperm were carefully placed right between the egg's outer shell and the egg itself. Then you had zona drilling and partial zona dissection, PZD, which were basically ways to physically create a little opening in that shell. These were all these really clever attempts to get around the natural barriers to fertilization. And wow, did things move fast. I mean, look at this timeline. In 1988, Jacques Cohen develops PZD. Just 1 year later, we get the very first pregnancy from a subzonal injection. And then, the true game changer. In 1992, Palermo and his team in Brussels announced the first live birth from intracytoplasmic sperm injection, or ICSI. That was it. That was the breakthrough everyone was waiting for. And the impact? It was just staggering. Think about it. From one single successful procedure in 1992 to over 2.5 million babies born just 20 years later. This just goes to show how ICSI went from this experimental, cutting-edge idea to a global standard of care for severe male infertility.
Okay, so now that we've got the backstory, let's get into the real meat of it. We're going to break down exactly why ICSI is used and talk about some of the really critical genetic implications that have to be considered before anyone starts treatment. So, here's the bottom line. ICSI became the go-to solution for conditions that before were pretty much untreatable. We're talking about severe male factor infertility, things like oligozoospermia, which is a really low sperm count, asthenozoospermia, which is poor motility, and teratozoospermia, which means the sperm have an abnormal shape. It's also absolutely essential when sperm has to be surgically retrieved for men with obstructions, and even for genetic diagnosis to make sure only sperm cells are involved in fertilization.
But, with a tool this powerful, you have to be incredibly careful. We know that subfertile men often have a higher rate of chromosomal issues, including things like Y chromosome microdeletions. There's also a known link between a condition called congenital absence of the vas deferens and mutations in the CFTR gene. Yep, the same gene involved in cystic fibrosis. And maybe most importantly, you could potentially pass infertility on to a male child. This is exactly why thorough genetic screening and professional counseling are absolutely non-negotiable.
All right, time to put on our virtual lab coats and step inside the embryology lab. We are going to walk through the incredibly meticulous process of ICSI, from the specialized gear to the delicate prep work, and of course, the injection itself.
This whole procedure demands insane precision, and that starts with the equipment. And there's a real trade-off in the systems labs use. On one hand, you have manual fluid-filled systems. They give you really direct, intuitive control, but they can get air bubbles or even leak. On the other hand, motorized systems are quick. They can even remember positions, but there can be a tiny delay in the response. Choosing the right setup is all about finding what works for a steady, efficient hand.
Okay, so the very first thing that has to happen is prepping the oocyte. After the embryologist removes the cloud of cumulus cells around it, they take a close look at each one. What are they looking for? A mature metaphase II oocyte. The telltale sign is a single, little extruded polar body, just like you see in image C. Only those mature eggs are ready for injection. The ones in stages A and B, nope, they're still immature and have to be set aside. The injection itself is like a highly choreographed ballet, and it happens in 6 steps. First, you select a single good-looking sperm and immobilize it with a flick to the tail. Then, you get the mature oocyte perfectly positioned on a holding pipette. The injection pipette comes in, pierces the outer shell, the zona pellucida, and then punctures the inner membrane, the ulemma. The sperm gets injected right into the cytoplasm, and then the pipette is carefully, gently withdrawn.
And these images, wow. They really capture that magic moment. On the left, you can see the injection pipette has just broken through the oocyte's defenses. The holding pipette on the left is keeping that egg perfectly still. Then on the right, the embryologist has moved the pipette in and is gently delivering that single sperm. I mean, the level of control required here is just phenomenal.
So, the next day is the big moment, the fertilization check, usually about 16 to 18 hours later. This diagram shows you what they might see. The one you want to see is image A, 2 clear pronuclei, one from the egg, one from the sperm, and 2 polar bodies. That's your perfect, normally fertilized zygote. Other outcomes, like 3 pronuclei or none at all, are considered abnormal.
But hey, the innovation didn't just stop with standard ICSI. Not by a long shot. The next big question was, how can we make this even better? How can we optimize the very first step, picking the absolute best sperm we can find?
One way is a technique called MAX. The science here is actually pretty cool. It's almost like a tiny magnetic filter for sperm. See, sperm that are starting to die, a process called apoptosis, show a specific marker on their surface. MAX uses tiny magnetic beads that are designed to stick to that marker. You run the sample through a magnetic column, the dying sperm get trapped, and only the healthy ones make it through. So what's the bottom line? By selecting for sperm with healthier DNA, this technique can potentially lead to better embryos and better outcomes.
Then you've got another huge leap forward, IMSI. The best way to think about it is ICSI, but with a ridiculously powerful microscope. Instead of the usual 400 times magnification, IMSI uses digital imaging to get up to over 6,000 times magnification. This lets the embryologist see the tiniest details of the sperm's internal structures in real time. This is especially useful for patients who have had repeated failures because it might improve fertilization and help create stronger blastocysts.
And just look at this slide. This really drives home what a difference that magnification makes. On the left, you've got a beautiful, morphologically normal sperm with its dimensions all measured out. But on the right, you see all these tiny defects, little bubbles in the head, weird shapes that would be totally invisible under a standard microscope. IMSI lets you select sperm on a whole other level.
What's really fascinating is that the same incredibly precise tools we use for fertilization can also be used after the embryo starts developing. So now we're going to look at other common micromanipulation procedures that are all about either helping the embryo implant or performing crucial genetic tests.
Okay, so it's really important we don't mix up 2 common procedures here, assisted hatching and embryo biopsy. Think of it this way. Assisted hatching is all about helping. Its only job is to create a small opening to help the embryo break out of its shell to implant. Embryo biopsy, on the other hand, is about testing. Its purpose is to actually remove a cell or 2 from the embryo so they can be analyzed genetically.
And these pictures show you exactly what we're talking about. Over on the left, you've got assisted hatching. A tiny needle with a mild acid is used to just dissolve a small patch of the zona, or shell. But on the right, that's a biopsy. See how the pipette has gone through the shell and is sucking up a single cell called a blastomere for genetic testing. 2 very different goals.
Now, when it comes to the biopsy itself, it can actually be done at 3 different times. You can biopsy the polar bodies, which only tells you about the mother's genetic contribution. You could do a cleavage stage biopsy on day 3, like we just saw. Or you can do what's pretty much become the modern standard, a blastocyst biopsy on day 5 or 6, where you take a few cells from the part that will become the placenta.
So when you step back and look at the whole journey, from those early fumbling experiments to today's high-tech, high-precision procedures, it's just incredible. Micromanipulation has given us a level of control over fertilization that was once pure science fiction. And it leaves us with a huge question, right? As these tools get even more powerful, even more refined, how do we as a society balance this amazing technological ability with the incredibly complex ethics of engineering the very first moments of a human life?